Hey everyone, welcome back to another deep dive. Today we're diving into something pretty fundamental.
Yep.
Is what makes you you. We're talking about genes.
Genes.
Yep, genes.
Yeah.
And to guide us through this super fascinating world, we'll be exploring Siddhartha Mukherjee's book, The Gene, an intimate history.
A fantastic book.
It really is. It's so much more than just like a straight science book.
Yeah.
It's full of stories,
Yeah.
surprising connections, and it just asks these questions about like what genes mean for each of us.
That's what I loved about it, really. It's not just like a textbook, you know?
Right.
Mukherjee's personal connection, his family history of mental illness.
Oh, yeah.
That really shines through. And you can tell it fueled his passion for understanding heredity. It just makes the whole topic feel so much more personal and relatable.
It does, definitely.
Yeah.
So, like, we all have this family history
We do.
that's shaped who we are in some way, right?
Yeah.
And this deep dive, I think, will help us kind of unpack that a little bit.
Let's unpack it.
Okay. Let's rewind way back, before scientists even knew what genes were.
Way back.
Like, can you even imagine trying to explain heredity without knowing about genes, without knowing the basic building blocks?
It's hard to even fathom. I mean, people had some really interesting theories back then. Like, take Aristotle, for example. He believed in this idea called the homunculus.
The what?
The homunculus. It's this idea that sperm contained a tiny, like, fully formed human just, like, waiting to unfold.
Wait, a tiny human, like, curled up inside each sperm.
Exactly. It's like a little miniature person ready to grow.
That's wild.
Mm.
But it shows you how long we've been fascinated by this whole question of where we come from and how we inherit traits.
Absolutely. Like, where do we come from? It's a fundamental question.
It really is.
And even after all these centuries of scientific progress, we're still wrestling with many of those same questions today, aren't we?
Yeah, we are. But let's jump ahead a bit. Let's talk about Charles Darwin, the father of evolutionary theory. He, like, revolutionized our understanding of how species change over time through natural selection. But even Darwin struggled to explain exactly how inheritance worked. Like, how are those traits actually passed down?
He was missing a key piece of the puzzle.
Which was?
The gene.
Exactly. Darwin saw these patterns of inheritance. But he just didn't have the tools to understand the mechanism behind them.
He needed that concept of the gene to really complete the picture.
He did. And speaking of patterns, Darwin's cousin, Francis Galton.
I've heard of him.
He was obsessed with measuring human variation.
Like, everything.
Practically everything, yeah. He meticulously recorded everything you can imagine. Height, weight, even like strength of pull and squeeze.
Strength of pull and squeeze. Like, what was he having people squeeze his hand?
I know, right? It sounds a bit funny now. But he even measured force of blow.
Force of blow. What was he doing? Like, measuring how hard people could punch? It does sound a bit out there, doesn't it?
Yeah. Like, can you imagine him with his little clipboard just running around measuring all these things?
Kind of amusing to picture. But Galton, he was actually onto something important.
Okay.
He recognized that people had all these measurable differences, right?
Yeah.
And he was convinced that those differences were largely inherited. And in fact, he even coined the term eugenics, which, we'll get into later. But
yeah, it has a
bit of a loaded history. Yeah.
Definitely.
Yeah.
But before we go down that road,
Okay.
let's talk about the person who actually cracked the code of inheritance.
Yeah.
Gregor Mendel,
Mm.
the monk with his pea plants.
Pea plants.
Pea plants. Who would thought? I mean, it doesn't sound that groundbreaking,
Right.
but his discoveries, truly, they were revolutionary.
Absolutely. It's amazing, isn't it?
Yeah.
This humble monk working in his garden basically laid the foundation for modern genetics.
Yeah.
His experiments with pea plants revealed some truly fundamental principles.
Oh, yeah, for sure.
I mean, principles that apply to all living things. For example, he found that traits, they're determined by these discrete units of information,
Okay.
which we now call genes.
So genes are like the instruction manual.
The blueprint.
Yeah, the blueprint for building an organism.
Exactly. And he figured out how they get passed down, too.
Oh.
He discovered that genes come in pairs.
Pairs.
Yep. These pairs are called alleles. And you inherit one allele from each parent.
So that's why you might have, like, your mom's eye color and your dad's hair color?
Exactly, because you're inheriting different combinations of alleles from your parents.
Okay, so I get the genes come from our parents.
Yeah.
But, like, how do they actually work? How do these instructions, these blueprints, get translated into, like, actual physical traits?
That's where Mendel's other big discovery comes in. The principle of independent assortment.
Okay, the what now?
Independent assortment. He found that different genes, they're inherited independently of each other, So, like, the gene for your eye color doesn't influence the gene for your hair color. They're passed down separately.
So it's not like mixing paint then.
No, not at all.
Like if you mix blue and yellow, you always get green.
Right. This shattered the old idea of blended inheritance, where
Ah,
people thought that traits were just like mixed together, like you're saying, like paint.
interesting. So instead of everything just blending together, we have these separate units of information.
Yes.
These genes, they can be combined in all sorts of different ways.
Like all sorts of different ways, yeah.
And that explains why siblings can look so different.
Exactly, because even though they share the same parents, they inherit different combinations of those genes.
It's like having separate blueprints for each trait.
Separate blueprints, yeah.
And those blueprints can be mixed and matched.
Creating all sorts of variations.
So cool. It's amazing how these seemingly simple experiments with pea plants could reveal such profound truths about heredity.
It really is.
I mean, who would have thought? But I'm sure a lot of people listening are thinking, okay, cool, pea plants, but what about me?
Right.
How do Mendel's laws apply to humans?
That's the question, isn't it? And that's where Mendel's work becomes really relevant to each of us.
Oh, how so?
Because his discoveries, they're the foundation for everything we know about how you inherited your mom's dimples or your dad's curly hair.
Wow.
It's the basic blueprint for understanding how traits are passed down through generations.
So even though Mendel didn't study humans directly,
He didn't, but
his work unlocked these secrets of inheritance for all living things.
it's pretty incredible when you think about it.
Yeah, pretty incredible for a guy hanging out in a garden with his peas.
Those peas were revolutionary.
But of course, the story of the gene doesn't end there, right?
Yeah.
There's still the question of what genes are actually made of.
Right, what's the physical stuff?
Yeah, and how do they physically carry all that information?
Yeah.
That's where the story takes a really dramatic turn. The discovery of DNA. The carrier of genetic information. We all know about the double helix.
Watson and Crick.
Yeah, Watson and Crick. But what led up to that iconic discovery? Like, how did they figure it out?
Well, after Mendel, the scientific community was buzzing with this question. What is a gene physically? We knew it carried information, but what was it actually made of?
It was in the materials.
Some scientists thought it might be protein, but others.
Others.
They suspected it might be this mysterious molecule called DNA. But how did they finally crack the code?
Yeah, how?
Well, the answer, it came from a rather unexpected source, pneumonia research.
Pneumonia. What does that have to do with genes?
It's a fascinating story. It starts with this British bacteriologist named Fred Griffith.
Okay.
Back in the 1920s, he was studying these two strains of pneumonia bacteria.
2 different strains.
Yeah, one was smooth and virulent.
Virulent, like dangerous.
Huh. Yeah, deadly. The other one was rough and harmless, and he injected this heat-killed virulent bacteria into mice.
So like the dangerous ones, but they were dead.
Right, they were dead. And the mice were fine. No pneumonia. But then he mixed the dead virulent bacteria with the live harmless ones.
Okay, so dead, dangerous bacteria mixed with live, harmless ones.
And injected this mix into the mice, and the mice died.
Dude got pneumonia.
They did, from the harmless strain.
Wait, so somehow the dead bacteria transformed the live ones? That's like something out of a horror movie.
It really is.
Like zombie bacteria.
Griffith called it a transforming principle.
A transforming principle.
Yeah, this mysterious substance from the dead bacteria,
Okay.
it somehow changed the live ones, made them deadly.
Wow. So genetic information could be transferred between cells.
It suggested that, yeah.
Yeah.
But no one knew what this transforming principle actually was.
It was a mystery.
A total mystery.
So we've got these mysterious dead bacteria transforming live ones.
Yeah.
And now the race is on to figure out what this transforming principle really is. This is where the story gets really juicy, right?
It does. And it takes us to Oswald Avery, a scientist working at the Rockefeller University. He spent years trying to purify this transforming principle.
Purify it.
Yeah, like isolate it. And guess what?
What?
It turned out to be DNA.
DNA.
DNA. But even Avery himself, he was hesitant to believe it at first.
Really?
Yeah, because so many scientists thought DNA was too simple a molecule to carry all the complex information of life.
They thought it was too simple.
Way too simple. But the evidence was pretty undeniable.
So now we know. Genes are made of DNA. But what does DNA actually look like? That's where Watson and Crick enter the picture, right? The race to discover the structure of DNA.
The double helix.
Yeah, that iconic image we all know.
One of the most famous images in science.
It is.
And this part of the story takes us to Cambridge, England,
Oh,
with this cast of characters. I mean, they were all brilliant.
Right.
And ambitious, too. You've got James Watson, this young, ambitious American.
Okay.
Francis Crick, this boisterous, quick-witted Brit.
Sounds like a fun duo.
They were quite a pair. Then there's Maurice Wilkins, a more reserved New Zealander, and Rosalind Franklin.
I've heard of her.
She was a brilliant chemist
Oh, yeah?
whose contributions were often overlooked.
Yeah, sadly.
But she was key to the whole story.
I sense some drama brewing here,
Yeah.
like a scientific showdown.
There was definitely some tension and competition. I mean, Wilkins and Franklin were using X-ray diffraction to study DNA, trying to capture those ghostly images
Those shadowy images, yeah.
that could reveal its structure. And at the same time, Linus Pauling was also in the race.
Didn't he win a couple of Nobel Prizes?
He did, a superstar chemist. And he actually proposed a triple helix model for DNA.
A triple helix. Was he close?
Not at all. It was a big blunder, actually.
Oh, wow. Even the best can be wrong, huh?
They can. But it shows how competitive the race was. And it lit a fire under Watson and Crick.
They knew they had to beat him.
They did. They knew they were close to cracking the code.
So what finally led them to the double helix? Was it Franklin's famous Photo 51?
Ah, Photo 51.
That X-ray image that, like, revealed the structure.
It was a crucial piece of the puzzle.
Yeah.
There's some debate, about how Watson and Crick actually obtained the image.
Oh, right.
And whether Franklin received proper credit for her work. But
Yeah, it's a whole other story.
it is, yeah. But it's clear that her work, along with Wilkins' data, played a key role in their discovery.
So picture Watson and Crick, right,
Mhm.
in their lab, building models, and, with wire and metal plates, just trying to fit all the pieces together.
Like a giant puzzle.
And then, boom, the double helix, this iconic twisted ladder with its paired bases like rungs.
Elegant, isn't it?
Yeah, it's beautiful. And it's suddenly revealed how DNA replicates, how it stores information, how it's passed on to the next generation.
It was a watershed moment in science.
It was.
Suddenly, genes weren't just these abstract units of inheritance. They were physical entities with a chemical structure that could be studied and eventually manipulated.
That's where we start to see the real power of understanding genes, I think. The ability to actually see the code of life and maybe even change it. But before we go there, I just want to take a moment and appreciate how far we've come in this deep dive. From Aristotle's tiny humans and sperm, to Mendel's pea plants, to Watson and Crick's double helix.
What a journey.
It's been a wild ride.
It has. And you know what's really mind-blowing?
What?
We're just at the beginning of understanding the complexities of the gene. I mean, there's so much more to explore.
Welcome back, everyone. So last time, we left off with this amazing image,
Yeah.
Watson and Crick unveiling the double helix structure of DNA.
It's beautiful, isn't it?
It is, like this elegant twisted ladder holding the code of life.
Yeah.
But I'm wondering, how does that code, that static code, actually translate into a living, breathing organism? How do we go from these A's, T's, G's, and C's to, well, us?
It's the big question, right?
It is.
It's like, okay, you've got the blueprint, right? But how do you actually build the house?
Yeah.
And this is where the central dogma of molecular biology comes in.
Central dogma.
Sounds serious.
It is. It's like the fundamental rule book for how information flows in a cell.
Okay.
So DNA gets transcribed into RNA, and then RNA gets translated into proteins.
Hold on. Transcription.
Yeah.
Translation. It's like we're decoding some secret message.
We kind of are. Think of DNA as this master cookbook,
Oh, a cookbook.
full of recipes,
Yeah.
right? But it's locked away in a library. And that library we can think of as the cell's nucleus. So to actually bake a cake, you need a copy of the recipe.
Right. You can't take the whole cookbook into the kitchen.
Exactly. So we transcribe the recipe.
Write it down.
Yeah, write it down on a separate piece of paper.
And that's RNA.
That's RNA. That's
No.
the copy of the recipe.
Kitchen
And then we take that RNA copy to the kitchen, where the real action happens.
being.
The ribosome, that's where the proteins are made. So RNA is like the courier, delivering the recipe to the baker.
Precisely. And in the kitchen, or the ribosome, that's where we translate those instructions into action.
Okay.
We gather all the ingredients,
Which are?
amino acids, the building blocks of proteins, and we assemble them step by step, following the recipe.
And what do we get?
A delicious cake. Or, in this case, a protein.
So every cell in my body has the same DNA cookbook,
Yeah, it does.
but they make different cakes. How does that work?
That's the amazing part. Not all genes are active all the time. Each cell has these ways of controlling which genes are turned on
Okay.
and used to make proteins. It's like, imagine you have bookmarks or flags in your cookbook,
To
right?
mark your favorite recipes.
Exactly. Highlighting the specific recipes that you need at that moment.
So it's not just about the code itself. It's about how that code is interpreted.
Yes, and
But,
used. It's about which genes are being expressed.
okay.
And this is where things get really, really intricate. Think about how different organs in your body have different functions.
Like your heart, your brain, your liver. They all do very different things.
Right, but they all have the same DNA.
So how do they specialize? Like, how does a heart cell know to be a heart cell?
It comes down to which genes are being expressed in each cell type.
So it's like each cell is reading different chapters of the DNA cookbook, depending on its job.
That's a great way to think about it. But how do they know which chapters to read?
Yeah, how?
That's a complex process.
I bet.
It's called gene regulation, and it involves a whole orchestra of molecules all working together.
Okay.
There are specific sequences in the DNA that act like switches.
Switches.
Yeah, like on and off switches for genes, turning them on or off.
Mm-hmm.
And these switches can be influenced by all sorts of things. Signals from inside the cell, signals from outside the cell.
So our genes aren't just static instructions. They're actually responding to their environment.
They are. It's like the genes are having a conversation with the world around them,
Wow.
constantly adjusting their activity based on what's going on.
That's pretty incredible. But it also brings us to another layer of complexity, right?
Another layer, yes.
Epigenetics.
Epigenetics.
I've heard that word, but I'm not really sure what it means.
Okay. So think of it this way. If DNA is the hardware, like the computer itself, then epigenetics is the software.
The programs that run on the computer.
Exactly. It's a way of controlling gene expression without changing the underlying DNA sequence. It's like, imagine you're adding sticky notes to your cookbook, right?
Sticky notes.
Highlighting certain sections, or even like crossing out others.
So those notes, they don't change the recipes themselves,
They don't.
but they influence how you use them.
Exactly.
Oh.
And that's what epigenetic marks do. They're like those sticky notes. They can fine-tune gene expression, make adjustments based on our experiences.
Our experiences. So, like, things that happen to us can actually affect how our genes work.
They can. And here's the really wild part. Some of these epigenetic changes can be passed down to future generations.
Wait a minute. So it's something that happened to my grandparents could potentially affect my genes.
It's possible.
Wow. That's incredible. And maybe a little bit scary too, right?
It's definitely mind-blowing. It makes you rethink the whole idea of inheritance. Like, what are we really passing on?
It's more than just our genes then.
It is. But before we get too deep into those philosophical implications, let's bring it back to something a little more concrete. Remember how we talked about how a single letter change in DNA can have a huge impact on a protein?
You mentioned sickle cell anemia.
Yes, sickle cell anemia. It's a perfect example of how one tiny change in that DNA recipe can really mess things up.
So one little typo can mess up the whole protein.
It can, and have a huge impact on a person's health. So
Yeah.
sickle cell anemia is caused by a single letter change in the DNA sequence that codes for hemoglobin.
Hemoglobin, that's the protein that carries oxygen in red blood cells, right?
That's it. And this single letter change causes the hemoglobin protein to fold incorrectly.
So instead of forming nice round red blood cells.
They become sickle-shaped, like a crescent moon.
Oh, right. I've seen pictures.
And those sickle-shaped cells, they can clog blood vessels, cause all sorts of serious health problems.
It's amazing how one tiny change at the molecular level can have such a huge effect on a person's life. It's a bit scary, actually.
It is, yeah.
It makes you wonder, if one little change can cause disease, what about all the other variations in our DNA?
Right, there are so many.
How do scientists even begin to make sense of all that information?
That's a great question. And it's what led to one of the most ambitious scientific projects in history, the Human Genome Project.
Ah, the Human Genome Project. I remember when that was announced. It seemed like science fiction, sequencing the entire human genome.
It was a huge undertaking.
What exactly did it involve?
Well, it was a massive international effort to map and sequence all 3 billion base pairs of human DNA.
3 billion. That's a lot of A's, T's, G's, and C's. What did we learn from all that sequencing?
Well, one of the most surprising findings was that humans have far fewer genes than scientists initially predicted.
Really? How many do we have?
About 20,000 protein-coding genes. which, it's not that many more than a tiny worm.
We're not that different from worms, genetically speaking.
In terms of gene number, not that different. It was kind of a humbling realization.
It is.
It suggested that complexity doesn't necessarily come from having more genes, but from how those genes are regulated and interact with each other.
It's like how a simple set of Lego bricks can be used to build incredibly complex structures, right?
A perfect analogy. It's all about how you put the pieces together.
Mm-hmm.
And the Human Genome Project also revealed these vast stretches of non-coding DNA.
Non-coding.
Yeah, it doesn't directly code for proteins. It used to be called junk DNA, but
Junk?
yeah, but now we're realizing it's not junk at all.
Oh.
Some of it plays a role in regulating gene expression, but much of it remains a mystery. It's like there are these hidden chapters in the book of life, and we're just starting to decipher them.
So even after sequencing the entire human genome, there's still so much we don't know.
Oh, absolutely. And that's what makes this field so exciting.
Yeah, there's still so much to discover. But what about the practical applications? Like, how has all this knowledge about our genome translated into real-world benefits?
That's where the field of genomic medicine comes in. By understanding our individual genetic variations, we can potentially tailor treatments, predict risks, even prevent diseases before they manifest.
So, like, personalized medicine, based on your unique genetic makeup.
Exactly. It's a paradigm shift in how we think about healthcare, moving from this one-size-fits-all approach to a more personalized preventative model.
It's like having a personalized health forecast.
Right.
But I imagine there are some ethical considerations, too.
Right?
Like, this power to understand and manipulate our genes raises some big questions.
Huge questions, like how do we use this knowledge responsibly? Who gets to decide?
Exactly.
And that's where we enter this brave new world of gene editing.
Gene editing. That's where things start to feel like science fiction. What are we talking about here? Like, what can we actually do?
Well, we're talking about technologies like CRISPR, which allow scientists to make very precise changes to DNA sequences.
So we can actually rewrite the code of life.
In a way, yes. It's like having this molecular scalpel that can cut and paste DNA with incredible precision.
Wow. And what can we do with that?
Well, it opens up the possibility of correcting genetic defects that cause diseases, like cystic fibrosis, sickle cell anemia, Huntington's disease.
There's like a genetic fix-it kit.
Yeah.
But are there any risks involved with this? I mean, messing with the code of life sounds pretty daunting.
You're right to be cautious. One of the biggest concerns is the potential for off-target effects. Like, CRISPR might accidentally cut DNA in the wrong places,
Oh, no.
potentially introduce new mutations, or even cause cancer. So while the potential benefits are huge, we need to proceed very carefully.
Yeah, definitely. And then there's this whole other ethical dimension, right? I mean, if we can edit genes to cure diseases, what's to stop us from using this technology for enhancement? Like to create designer babies with specific traits. That feels like crossing a line.
It's a slippery slope, for sure. We need to have some serious conversations about where to draw the line. How do we balance the potential benefits with the responsibility to use this technology ethically?
It's clear that the story of the gene, it's about so much more than just science.
It's about our humanity.
It touches on our deepest values, our fears, our hopes for the future.
And it raises some really big questions, questions that we as a society need to grapple with.
From Mendel's pea plants to the mind-blowing possibilities of gene editing, we've come a long way in our understanding of heredity.
And the journey is far from over. Every day, scientists are making new discoveries.
So what does this all mean for us? For each of us as individuals navigating this, like, brave new world of genetic knowledge, how do we make sense of the fact that our genes, while not our destiny, do profoundly influence who we are?
That's a question we'll be exploring in more depth in the final part of our deep dive.
Welcome back to our deep dive into the gene. I don't know about you, but my head is spinning a little.
It's a lot to take in.
It is. I mean, we've gone from these ancient theories about tiny humans curled up in sperm to, like, mapping the entire human genome. And now we're talking about editing the code of life itself. It's mind-boggling.
It really is.
But after all of this, I'm still kind of stuck on that age-old question. Nature versus nurture. Like, how much of who we are is actually determined by our genes? And how much is shaped by, our environment and experiences?
It's the million-dollar question, isn't it? And our understanding keeps evolving. As we've learned more about genes, we've realized that they're not just these static blueprints, dictating our fate.
Right.
They're dynamic. They interact with our environment, with those epigenetic modifications we talked about. They fine-tune gene expression.
Right. So our genes are having this ongoing conversation with the world around us.
Exactly. It's a two-way street.
But those twin studies we talked about earlier, they still make me wonder, If genes play such a big role, why aren't identical twins, with their identical DNA exactly alike in every way?
Ah, that's a great point. It highlights the role of chance and individual experience. Think about this study. Scientists took genetically identical worms, right?
Okay, worms.
Raised them in the exact same controlled environment. Everything was the same, but they still developed distinct traits.
They did. But how's that possible if their genes and environment are the same?
Well, researchers found that random fluctuations in gene expression, like these tiny variations in how genes are turned on and off, those had a big impact.
So even at the most basic level, there's this element of randomness. It's like maybe our genes provide the ingredients, but the final recipe is influenced by a sprinkle of chance.
That's a great way to put it. And as we move from, simple organisms like worms to more complex beings like humans, the interplay of genes, environment, and chance, it just gets more intricate.
Right, because we have so many experiences that shape us.
Exactly. Our choices, our relationships, the challenges we face, all of those things, they can influence gene expression, and they contribute to our individual journeys.
It's both mind-boggling and kind of liberating, isn't it? To think that even with identical DNA, you and your twin could lead very different lives.
Absolutely. Our genes provide a foundation, but they don't define our destiny.
We have the power to make choices, to shape our own path.
We have agency.
So it's not really nature versus nurture then, is it? It's more like they're partners in this intricate dance that shapes who we become.
I love that analogy, a dance. And this realization, it has profound implications for how we view ourselves, our health, our place in the world.
Yeah, it emphasizes that each person's story is unique, shaped by this, incredibly complex mix of factors. We can't just reduce someone down to their genes, because those genes, they're just one part of a much bigger, more fascinating story.
Exactly. And it also highlights the importance of empathy, I think, and understanding. We need to appreciate that everyone's journey is influenced by this unique combination of things.
It makes you realize that the story of the gene is ultimately a story about our shared humanity.
It's about the incredible diversity and resilience of our species.
And knowing that fills me with a sense of awe, for the sheer complexity and beauty of life.
I agree.
It's humbling
Yes.
to realize how much we've learned, but also how much there still is to discover.
We've just scratched the surface.
We have.
Uh,
It
the-
feels like we've only just begun to understand the gene and the journey ahead. It's full of exciting possibilities, but it also raises some pretty profound questions,
It does.
Like if we could edit our genes to eliminate disease, would we be losing something essential about being human?
It's a question worth pondering.
Would we be erasing the very variations that make us unique? The things that drive our evolution, that fuel our creativity and
It's
resilience.
a delicate balance.
Yeah.
And we need to have these conversations. As we venture further into this uncharted territory, how do we balance the potential benefits with the responsibility to preserve our humanity?
It's a challenging conversation,
It is.
but it's a crucial one as we continue to unlock the secrets of the gene. Well, I want to thank you for joining us on this incredible journey through the world of the gene. It's been a wild ride.
It has.
I hope you learned some things, challenged some assumptions, and maybe you came away with a renewed sense of wonder about the, incredible tapestry of life. And remember, the exploration doesn't end here. Keep learning, keep questioning, and keep diving deep.